Great. Then let's go ahead and get started. Welcome everyone to the Thursday DMICE Informatics Research Conference at our normal time for this installment. And as often happens at the end of the academic term, we have a number of defenses, proposal defenses, and so forth. Today, we are having a PhD dissertation proposal defense from Nate Evans, who is known to most, if not all, of the attendees at the meeting. Nate is a PhD candidate in the Bioinformatics and Computational Biomedicine major. He previously to OHSU studied biological physics at the University of Washington and was employed as a mechanical engineer developing lung cancer diagnostics. His research goals are to develop robust drug response models to aid decision-making in precision oncology. His current work has broad translational application in drug development, combination therapy prediction, clinical patient response prediction, and patient therapy toxicity prediction. So he's defending his dissertation proposal today. We'll get feedback from the audience. We after his talk, we'll open it up to everyone here. Then the public will leave, and leaving behind his dissertation committee if they have any further questioning or suggestions that came from the public talk for him. And if he passes that part of the if he passes the proposal defense, then he moves on to do his dissertation work, and we will hear back from him some number of months down the road where he'll come to defend his dissertation. So I'm going to turn it over to Nate.
Thanks, Bill. I appreciate that introduction. First of all, can everyone see the right screen, presentation mode, and hear me?
Yeah, looks good to me.
Perfect. All right. So the title for today's proposal defense is Improving Prioritization of Preclinical Drug Screening Based on Genetic Background by Using Bayesian Optimization in a Continuous Distributed Representation. There we go. So to frame the talk today, I want to start with this research question where if we're given a cancer omics, how can we identify an optimal treatment? And so there's a number of ways to approach a question like this. However, from a computational perspective, we often start by asking what data do we have available to us? And so there's been a ton of work done in the molecular characterization of cancers. And these are stored in repositories like the Cancer Genome Atlas and the Cancer Cell Line Encyclopedia, or the Gene Expression Omnibus. And then we've also done work in characterizing oncology drugs. This is commonly done by their molecular structure or by which proteins the drug binds to and how it changes protein behavior. And then kind of in parallel to this, there's been pretty remarkable work done in measuring the functional response of different cancer model systems. And so these model systems could be cell lines or mouse models. And when I say functional response, a common data type for this would be like dose response curves or cell viability. And so given this data, we can think about building these predictive models that take in your cancer omics and drug features and tries to predict the functional response of these model systems. And it's really important to remember that when we build these models, we need to make sure that our results are going to generalize to patients because that's where we get our impact. And this is going to kind of frame the talk for today. And so as oncology therapeutics currently stand, we have a major issue with drug resistance. And so there was this paper that cited in 2017 of about 0.6 million cancer deaths. 90% of those were attributed to drug resistance. And this problem that we see is especially common in targeted monotherapies, where initially we have pretty good response. The tumor is sensitive and responds well. However, over time, we see the tumor acquire resistance and stop responding to the drug. And we believe that one of the reasons for this is this tumor heterogeneity, where there's multiple clones, cancer clones, some of which may be sensitive to a drug and some may be resistant. And so the drug essentially acts as this selective pressure that evolves resistance. And so one of the ways to address this is by using combinatorial therapeutics, where we use more than one drug. And, this might target more of the cancer clones, or it could also result in what we call synergistic behavior, where the effect of the drugs together are much better than either single agent. Now, we also see a problem with in adjuvant therapy where the side effects can impact the quality of life of the patients. And so in extreme cases, this can result in discontinuation, which can affect the overall outcome of our patients. And so if we can improve the toxicity profiles of these drugs, then I think we can also address that problem. And so our goal here is that we want to get more and better therapeutics into the hands of clinicians where they can do some good for patient treatment. However, there's this problem as well in the research pipeline, specifically in getting these oncology drugs through FDA trials. And so for monotherapies that enter clinical trials, very few of them actually get FDA approval. And as a quick overview, there's three primary phases of an FDA trial, and the first one addresses the toxicity of a drug. And then the second phase looks at if the drug is effective at all in treating the given disease. And then the phase 3, looks at comparing to the standard of care. So is it any better than what we already have? And so this 3.4% of monotherapies that actually enter are approved. And so we have this real bottleneck in the research pipeline there, and we want to try to improve that in some way. And so we might just start by asking, why do we need to build computational models to predict these drug combinatorics at all? Why don't we just measure everything at the bench and pick the drug combinations that work the best? And the problem with this is that this combinatorics problem becomes intractable quite rapidly. And then on top of that, there's two additional axes to consider. One, that genomic differences can change how a tumor responds to a drug, so the drug sensitivity. And then we can also prioritize concentration-specific combinations. And so this becomes a really large search space. And as an example, let's say that we want to explore pairs, so 2 drug combinations of 10 drugs, and we're going to measure 7 concentrations, which is fairly common with a dose response curve. And we want to do this with 1,000 cell lines. And so in this example, to measure every combination, it would require 2.2 million assays. Now, if we bump that up to 100 drugs, that's going to be 242 million. And if we bump that up to 1,000 drugs, that's 24 billion. And so I think it's easy to recognize that we really can't measure everything. And so this is a bit of a shift to how can we efficiently explore this drug space to identify those therapeutic combinations without having to measure all of them. Now, let's take a quick step back and imagine that we've come up with this computationally guided research methodology that takes in cancer omics or a genetic background and produces an optimal therapeutic, which could then go on to FDA trials. And the question is, what would success look like here? And so first, for any inputs, we want to generate some output. We don't want to fail to identify a therapeutic for some tumor. We also want to understand why we prioritize the drugs that we do. So this comes down to the interpretability of our methodology. And because a lot of the available data comes from model systems, we need to make sure that whatever we prioritize generalizes to patients. That's where we get our translational impact. And we want to minimize those toxicities, both for getting drugs through phase 1 FDA trials and for the problems with side effects during adjuvant therapies. And then finally, there's these pragmatic bounds in research where if we want to have translational impact, we really have to make this fast, cheap, and efficient. And so we want that. And for context, this is going to be a preclinical step, which would go into clinical trials. So there's been a lot of work in drug response modeling, and then deep learning is a kind of a subset of machine learning commonly used for this. However, there's still a number of limitations. The first one I'm bringing up is what we call inflexible modeling. And I'm not sure that's quite the right term, but it's this idea that we have difficulty in incorporating different data types or different forms of data like prior knowledge or structural information. And so addressing that can really be a boon to the field. We also, in drug response, we often have small data sets, and this comes to, compared to the massive search space, we can't measure everything. And so we have sparsity, and this translates to what we call epistemic uncertainty, where we may just not have data to characterize the underlying model. And then there's a second form of uncertainty when we have really noisy data types. And this is called aleatoric uncertainty. And we commonly see this with genomic or omic measurements where we can have significant noise, or with drug response metrics like cell viability that has significant noise between replicates or between data sets that can have pretty poor concordance. Now, with deep learning, we commonly think of these models as black box that have inputs that come in and predict some output, and they're really powerful. However, they also often suffer in their interpretability. And so there's a number of more recent methods for addressing this, specifically in drug response, but I think we can do better as well. And so this is a goal of ours. And then finally, there's this concept of parameter sharing, and to explain that, it's easiest done with convolutional neural nets. And this method learns what they call a convolution or a kernel, which can be applied to a contiguous region of pixels to extract some feature. And what a method like this does is take advantage of the structural information in the image, more specifically that nearby pixels are spatially similar. And so in other domains, when we can recognize this structural information, we can also take advantage of parameter sharing, and that can be wildly successful or advantageous to model performance. Now, I touched on that we have significant uncertainty in drug response due to either we don't have enough data, or the data that we do have is noisy. And so to address a problem like that, we can use this a method called uncertainty quantification, where we train a probabilistic function to explicitly recognize where we have enough supporting evidence to make a prediction. And we can do this with what we call probabilistic predictions. And so to explain what those are, let's imagine that in this figure at right, we make a prediction at x equals 7. And so the prediction of y is centered around 0. However, it's a probability distribution over a range of values. And so for this example, our probability distribution is very narrow, right around 0. And so we have high confidence that the value of y is close to 0. However, if we make a prediction at x equals -7, we have a really significant spread on the prediction of y. And so because of that, we have very low confidence. It could be -5, it could be 5. We really can't say. And so methods like this are really advantageous when we want to use our predictions for decision making, or if we want to reduce the false positives. And I really like this concept that we're letting the model say, I don't know, instead of forcing it to make a prediction. And this becomes advantageous later. Now, the methods I propose are going to involve what's called a variational autoencoder. So to explain what that is, I want to start with an autoencoder. And this is an unsupervised form of neural networks. And what we do is we take some input data in and we bottleneck our neural network. And then we use this reduced dimensionally latent space in the bottleneck to try to reconstruct the original data. And so the loss function just becomes the error between the reconstructed and the input data. And so this actually has a lot of similarity to principal components analysis. However, PCA is linear. And so if we think about projecting our data onto one of these linear PCA components, we would project the blue dots onto the red line. That would be PCA. However, autoencoder learns this nonlinear component, and we would project the data onto that. And so I think it's pretty straightforward to recognize that in situations where we have complex nonlinear data, the autoencoders can be really, really advantageous. Now, variational autoencoders, they have a similar architecture, but a different task. Their goal is actually to model the probability distribution of a data set or of an observation. And so they do that by taking in some input data, like the autoencoder, and bottlenecking the network down to some reduced dimensionality. However, rather than just predicting a single value for that latent space, it predicts both a mean value and a variance value. And now we can make the assumption that our data is normal in the latent space, and we can sample from a normal distribution. And so what we've done is inherently characterize or represent our input data in this probabilistic way, which is pretty cool. And so to build some intuition on how this method works, let's imagine that we've trained a variational autoencoder on the MNIST dataset. And MNIST is images of hand-drawn numbers. And so if we've trained this variational autoencoder, we can sample from the latent space. And so this new image below is if we were to sample from some region of the latent space, the number is the image we would get from the decoder. And so you can see that this latent space does a really nice job of representing the images in 2 dimensions. Not just that, but it's done a good job of blending one number to a next and quantifying any ambiguity or uncertainty. So for instance, down here, where 1s, 8s, and 7 all blend, there's a lot of ambiguity, as well as up here with the 0s and 7s. Now we can think of this from the other side, where if we take some input data in, so imagine we feed that image of the 2 into our network, then we produce this probabilistic representation in the latent space. So it's going to predict some normal distribution down here in the 2s. And so that's pretty neat. However, let's imagine that our input data is, instead of just being a clear 2, let's pretend that we can't really tell what it is. Maybe it's a 2, maybe it's a 3. Well, in that case, our encoder function can predict a value overlapping both 2 and 3s. And so we've now quantified our uncertainty that the input data could be a 2, it could be a 3. We're not totally sure. And so that's kind of that uncertainty quantification that I like so much. Now, these are really powerful methods, and they work well in a number of tasks, especially low data volume tasks. But one of the reasons that I really like them is this idea of learning disentangled variables, which we can think of as independent variables. And so that's shown in this figure at right, where this was taken from a variational autoencoder trained on images of celebrities' faces. And what it learned is that if you sample from a higher value of that x-axis latent variable, then you can add or remove glasses from the face. Whereas if you sample from the y-axis latent variable, you can change the type of glasses. And so this is really cool because they're independent variables, they're very interpretable. And as you're adding glasses, you're not also changing hair color. So this really does add a lot of functionality and interpretability to our models. So here are my aims overview. From a high level, aim 1 is just to build a drug response model. we're going to employ a variational autoencoder. And there's a little bit of nuance in how we set up our encoder architecture that'll operate on graph ontologies. And then we'll train this model on cell line data where we use unperturbed omic features to predict both perturbed expression features and those classical metrics of drug response like cell viability. And then in AIM2, we're going to shift away from just trying to predict an optimal therapeutic. And we're going to make this assumption that we probably don't have enough data to have high confidence in any of these combinatorial predictions. However, we can explore efficiently. And so we're going to think about reframing this drug combination prediction as an optimization problem. And we'll go in more depth on how we do that. So this is the official specific aims 1. And the key points to take out from here is that we're going to train this variational autoencoder to predict drug response and perturbed omics from unperturbed omics. Now, before I get into the details here, I want to introduce this prior art, and it's a model called Dr. VAE, and they have a really nice graphic framing the problem. This is actually a very similar task and architecture to the AIM that I've proposed. However, it has a number of limitations. First of all, they use fully connected networks for their encoder and decoders, and so this doesn't take advantage of any structural or prior knowledge. It doesn't use parameter sharing, and I think that's a bit of a limitation. They also only trained it on the Lynx L1000 dataset, which is RNA expression features. And so I think we can do better on a number of other data sets. And so we'll benchmark against this model to see if we're improving. This is the architecture of my proposed model. And the main thing to note here is that I'm proposing that we use 2 generative or probabilistic layers. So Z1 and Z2 will both be probabilistic, where Z1 represents cell state, and Z2 will represent the functional response or perhaps the cell viability. And the reason I think this is really important is that you could imagine if we have absolute certainty in cell state in Z1, we could still have very significant uncertainty in the functional response of that cell state. And so by separating these two, I think we're really going to improve the calibration of our predictions. Now, each one of these encoder or decoder functions are just parameterized by neural networks. And when I refer to them throughout the talk, I'll call them the cell state encoder or the perturbation decoder or the functional decoders. And for the next couple slides, I'm going to walk through a toy example showing how I want to set up my encoder architecture, because there's some nuance there. And so first, this encoder architecture is going to operate on some graph ontologies, specifically protein-protein interaction graphs, where nodes represent proteins and the edges represent protein interactions. And so the first thing we can do is overlay our omic features as node attributes. However, one thing to note is that we often in these omic data sets, we have sparsity in which features are actually measured. And so because of that, we'll have to address it, and we can do that with imputing. And there's some really nice methods that have already been developed specific to imputation on graphs. And so that fits very nicely into this. And then the next step, well, first, let's recognize that this base graph that I've produced, it essentially represents a cell line's interactome, which proteins are interacting with each other. However, we know that from cell line to cell line, there's pretty significant differences in the interactome, that these maybe in one cell line, 2 proteins do interact and in another they don't. And so to address this variability, I'm proposing I use what's called an attention mechanism to predict individual observation interactomes. And so this shifts our undirected, unweighted network into a directed weighted network. And this is also really nice because to some extent, we're borrowing an idea from pharmacology, specifically called coupling, where in a signal transduction network, we can think of the signal or the information either growing or decaying due to the number of inputs and the outputs, or how two proteins might interact. And so in this methodology, I'm interpreting these edge weights as what I call coupling coefficients, because they're kind of implicitly bottlenecking the information they can pass between two genes. And by doing this, we can introduce a really novel way of encoding our drug perturbation. And so if some drug targets a given protein, we can think about modifying the edges to or from that protein to encode the actual perturbation. And so notice that those edges have decreased. So maybe if that drug was an inhibitor, that would be a good encoding. Now, the last step is to ask how we actually learn from this data. How do we actually recognize the complex patterns that we can make predictions from? And to do that, we're going to use something called graph convolutions. And to walk through a toy example of how a graph convolution works, we'll just look at one node. So the next layer node value here is going to be the sum of all of its neighbors, including its self edge weight, which is that one. And then we have the edge weight of incoming edges times some function of the neighboring node values. And we put that in a nonlinearity, and that becomes the next node layer. Notice that in this case, I only care about edges coming into my node. That's a hyperparameter that we might choose. And so really what this is doing is allowing information to pass from node to node, and we can extract more complex features. But it does it in a way that constrains the features' interactions. So only edges that exist, so that when edges exist, we allow those features to interact. And that comes down to prior knowledge. And so that toy model that I just walked through can be summed up in this image. And I'm not going to spend a whole lot of time here except to say that we can represent this a little more cleanly, and we can revisit if there are more questions. So to revisit this idea of encoding our drug perturbation as edge modifiers, let's focus in on kinase inhibitors. And so the signal through a kinase inhibitor is mediated by its upstream proteins and the binding of a phosphate, which is going to stabilize the active conformation and produce some downstream signaling. And so we can model that by this coupling coefficient, which I'm interpreting as those edge weights in my previous model. Now, in the presence of a kinase inhibitor, that drug binds to the kinase, it stabilizes the inactive conformation, and we reduce the signal out. And so the question now becomes, how do we change that coupling coefficient as a function of drug features? Now, this is really nice because it's mechanistic and interpretable. However, there are some limitations in that we have to know which targets a drug binds to, and we have to know how tightly the drug binds to the target. We also need to know that how the drug changes protein behavior. So when it binds, does it activate the protein or does it inhibit the protein? And that's very important as well. And then finally, because we're encoding this as an edge modification, we need to know gene-gene interaction knowledge, and that needs to be accurate. So I've done some preliminary work here, and starting with the definition of binding affinity, this equation here, where KD is binding affinity, we can derive this function where C star represents the drug perturbed coupling coefficient. And this is in terms of the drug concentration and the drug binding affinity to some specific drug target. So moving on to what data types can we use to train a model like this? I want to revisit basically the elements or the paths of information through the model. And so we can create these unperturbed cell state representations. That would be the blue circles there. Or we can create a perturbed cell state representation using our drug perturbation method. And then using these latent representations, we can predict cell viability, or we can predict the expression of those cell states. So now the routes of data or of information through this model dictate what data types we can train the model with. And so first we can think about the unsupervised approach, where we use unperturbed omics to predict unperturbed expression. This is probably the most classical form of VAE training. We also, by definition, know that an unperturbed cell line should have cell viability of 1, so we can annotate that. And then we can also use this drug perturbation encoding to create a perturbed cell state, and then we can predict perturbed expression features. So this lets us take advantage of data sets like the Lynx L1000, which measures perturbed expression, or the Cancer Perturbed Proteomics Atlas, which measures perturbed proteins. And then last, we have this classical form of drug response training where we predict cell state and then we use that to predict cell viability or drug response metrics. And so this is really nice because these 4 different forms or routes represent different data types that we can use to train the model. And this addresses that low data volume that we often see in drug response. Now, I'm not going to spend a whole lot of time on this table. The key pieces are that I've identified a number of different data sets with fallback options to use in training this model. And I think there's a lot of work to be done into choosing and optimizing these, how we can incorporate them. So to evaluate a model like this, we can use holdout test sets to evaluate different metrics of performance. However, I've broken this down into a number of really critical questions that I think we need answered. The first one being, are our predictions well? You remember back to my symposium, calibration is this metric of uncertainty quantification performance. And so for that, we can use a metric like expected calibration error. But the reason I care about this is that because in AIM2, the prediction calibration becomes very important. Now for the second question, we want to make sure that our latent representation is good at characterizing the cell state. And so we can evaluate that with how accurately can we predict perturbed expression. And for this, we can actually benchmark against Dr. VAE, as it has a very similar architecture and task. We also want to make sure that we can predict drug response well. Once again, we can benchmark against Dr. VAE. However, there's 2 additional models that I would consider. One called Drug Cell from the Idacre lab, and another one called Deep CDR that has pretty admirable performance right now. These last 2, we want to make sure that we can interpret our predictions. And the Idacre lab has done some really incredible work on how you might do this in deep learning. And so we can co-opt a few of those ideas. And then last, because we're going to use this continuous latent representation of our model in AIM2, we want to make sure that this is smooth and informative. And there's some qualitative methods we can use to try to address that. So moving on to AIM2, we're going to shift from, instead of just trying to predict an optimal therapeutic, let's make the assumption we don't have enough data to do that. And instead, let's ask, how do we most efficiently explore this massive drug space? And so we're going to do this by using the latent representation of cell state from our AIM1 model, or alternatively, we can use Dr. VAE if it outperforms. And we're going to let the model request data where it'll be most informative or most useful. And we believe that this will converge to an optimal therapeutic after some chain of prioritizations. And so I want to introduce this prior art, which has a very similar task in a different domain the goal of this model was to optimize chemical design. And so what they did is they trained a variational autoencoder to which took smiles or molecular structure as input and created this continuous molecular representation. But the smiles is just a string representation of drug structure or of chemical structure. And so once they've created this continuous representation, they were able to train classifiers to predict some property that they might want to optimize over. And so they've shown that in the graphic at right, where the global maximum here represents an optimal property. And so if they find that global maximum and then locate the corresponding latent space, and they can decode that back into the smiles representation, back into a testable drug structure. And so this is a really nice way of taking a very high dimensional, difficult problem, creating a low dimensional representation of it, and then optimizing in that low dimensional representation. And we're going to try to do the same task, however, in drug combination prediction. And so to understand this, we have to first kind of go over this idea of Bayesian optimization, which is the method we'll use to actually explore. And this is a method commonly used in hyperparameter optimization, where you can imagine if you're trying to build a random forest with the highest accuracy, you have to choose the hyperparameter, the number of trees in that random forest. And depending on what you choose, can change the accuracy of your model after you've trained it. And let's imagine that this is a really big model and it takes a day to train. Well, in this case, we can't actually measure all possible K values. And so to do this really efficiently, we can train a probabilistic, we call this a surrogate function, to predict the model performance from the number of trees in the random forest. And that's represented with that purple and blue line in the figure. The black line represents the true model performance. And so using this probabilistic function, we can generate what we call an acquisition function, and that's this expected improvement here. A really intuitive example of an acquisition function would just be the upper confidence bound. And so using the acquisition function guides you which K values we should next test to most efficiently explore this space. And so it's really nice, really clever method of optimizing hyperparameter optimization, or essentially any black box optimization. And so we're going to use the same method. However, we want to use it for drug combination exploration. So the x-axis is going to be the latent representation that we would take from our AIM-1 model, and the y-axis is this metric of drug response. And in this figure, we could imagine that it's 1 minus cell viability. So we're trying to look for a global maximum here, which represents an optimal therapeutic. Now, if we were just to use the upper confidence bound as our acquisition function, we would identify this red line, identifying that that's where we should, the model should ask for more data. And so using this latent representation, we can decode it back into a set of drug combinations, which we can test at the bench. Now, I've shown this figure before, but for the sake of simplicity, let's, for the moment, forget about uncertainty and just represent that our encoder function can create some point in the latent space that represents the cell state representation. Also, with some drug, we can create a perturbed cell state representation. And you can imagine that for any unperturbed cell, we can create many, many different perturbed cell states. However, not all perturbed cell states. And this could be done with different drugs or a combination of drugs, or maybe a different concentration of the same drug. Now, if we imagine all possible perturbations that could happen on a cell line and all the resulting cell states that would come from that, then we get this subspace that represents the perturbable states of a given cell line. And so by calculating this, what we've done is condition on a set of omic features representing a cell line. And that becomes the first step in our algorithm for AIM2. The second step is to use our Bayesian optimization methods that we went over to identify this candidate region, representing that it's an optimal region to explore in terms of this therapeutic metric that we want to optimize. And then the last step is to take the candidate region and decode that back into testable drug combinations. Now, everything that I've proposed up to this point has used cell viability as the metric that we're trying to optimize. And the problem is that cell viability is essentially cytotoxicity, and there's nothing keeping our algorithm from prioritizing very high concentrations of a drug or multiple drugs in combination that essentially would just kill all cell lines. And the problem with that is that it's essentially, it would be a very toxic treatment to any patient, and we would never use it. And so we can address this by imposing some constraints. The first and most naive constraint would be to control the sum of drug concentrations. Alternatively, we could calculate the average cytotoxic response of some given candidate region over either a set of control cell lines or potentially all cell lines. And then the third one is if we go back to this idea that we have these subspaces that represent the perturbable region of a cell line, then we can imagine two subspaces. One subspace that represents the perturbable region of the cell line that we're trying to prioritize for, and then another subspace which represents the perturbable region of all other cell lines. And if those two subspaces don't overlap or don't entirely overlap, then we can infer a selective response, not necessarily a selective cytotoxic response, but at least a cell state differences. And then finally, the fourth one is this idea of prior knowledge, that if we know combinations that are toxic, we could actually use the latent space to train classifiers to predict the toxicity. And there's already been some really nice work done on incorporating such classifiers into Bayesian optimization as constraints. And so that's actually a really exciting avenue. However, it could be challenging to build these toxicity data sets, especially in cell lines. Now, validation of an algorithm like this can be a little challenging. However, there's both a computational route and kind of a wet lab route. So for the first question, does our algorithm prioritize effective candidates? The computational method for this would be to withhold known effective observations and then look at their rank in our prioritization. And what we would expect to see is that they're ranked fairly high, that we should explore these regions. Alternatively, we could also do bench testing of the prioritized candidates. And we would want to do this for both positive and negative to make sure that we're doing this well. Also, because the way Bayesian optimization is set up, we don't necessarily assume that every prediction or every prioritization is going to be a super effective candidate. Remember, we're trying to converge to that global maximum. And so really we need to test this chain of prioritizations. And so we could do that with bench testing if we iteratively test multiple drug combinations. However, we could also do this with a simulation where we make up fake data and we withhold the majority of it from the data and then let it iteratively request data as it makes the predictions. And what we'd expect to see is convergence to the global maximum. And then the last validation step would be to make sure that we prioritize low toxicity combinations. And so we can look for selective cytotoxicity in the cell lines of interest compared to control cell lines. or we could potentially look at how well the combinations are handled in mouse models. Now, to circle back to impact of research like this, I think that if it's well done, this could really change how we approach oncology drug screening. And there's a number of ways that's going to affect, patient impact or translational impact. The first of which, if we can make this a efficient and rapid method and identify these combinations, we can just put more drugs into the pipeline, which is obviously a benefit. If we can improve the toxicity profiles of these drugs, then via our toxicity constraints that I touched on, then I think we can also bump up the number of drugs that make it through phase 1. If our convergence algorithm identifies more optimal therapeutics, then that's also going to bump up the numbers for phase 2 and phase 3, which ultimately puts more drugs into the hands of clinicians where they can help patients. And so I think that's pretty exciting, and if done well, could have a lot of impact. And with that, I'd like to acknowledge my advisor and DAC and everyone who helped me prepare for this talk and put the ideas together. And I'll take questions.
Great, thanks for a nice talk. People who have questions can either unmute and speak up or type into the chat.
Nate, this is Shannon. Great job. One of the things that, just kind of going to your last slide, we think about is, how soon is now? Are we ready, for this? So if your dissertation is successful, what do you think some of the next steps would be? We're thinking about actually having something that really could be put into a pipeline.
Yeah. I think this would work well integrated into like a bench, like a preclinical screening lab, where this would guide exploration. It's a good question, and I'm not I don't have a great answer for you.
Your answer is fine. It's just a thought. It's something for you to think about. We talk a lot about the methodology development, but if we actually want to have clinical impact or translational impact, it's always good to kind of look to the horizons too, in terms of thinking about it. And you've thought a lot about this with the optimization piece and the efficacy piece, so you're already thinking about these metrics, but just something to keep in mind, right? What is the kind of chasm between here and implementation?
Right.
So Nate, this is Shubo. Can you go back to where you introduced the VAE?
This one, right?
Okay, next slide. How you are going to use this? Okay, keep going.
This one?
Keep going. There's one slide where you wanted your latent variable 1 corresponds to 1 versus 2 corresponds to different, maybe 15, page 15.
Oh, the Bayesian optimization slide?
No, before that.
Okay.
Okay, maybe, let's see. 11, 12, 13. What about 15? Okay, so here, page 15, Z1 and Z2. So you said you have 2 latent variables with Z1 and Z2, and hopefully Z1 corresponds to the cell state representation, and Z2 would be the functional response representation. My question is that how can you control what latent variable corresponds to what? This is to be discovered from the data. You can't really dictate how many Z's are there. Well, you could somewhat dictate how many Z's are, but you can't tell or dictate which Z corresponds to what. The MNIST data, for instance, and then the face image with hairstyle and sunglasses, those happen to be the latent variables that was discovered from the data. You can't really go, I'm not sure what mechanism would be to go tell can you find a latent variable that corresponds to if there's somebody's wearing a sunglass? This is my point.
Right. So interpretation of latent variables would kind of be done after we've trained the model, that we'd have to go
Yes.
in and evaluate each
Right.
one.
Right.
So one way that we can separate Z1 and Z2, though, or is at least how I'm imagining it, is we can train. You could almost think about training Z1 with, just cut off everything to the right of Z1, and we would train the left half of this model with either the unsupervised method with just expression features, or we could predict this perturbed expression. And so in this case, the primary goal here is to reduce expression or omic features down into a really compressed latent variable, which I think would be a decent argument that you could say is a representative of cell state. And a naive approach would be to train this first, freeze all the parameters, and then train Z2 second. I probably won't do it that way, but I think that would be very interpretable as, or at least more interpretable as Z1 being cell state. That's how I'm rationalizing.
You have to, after training is done, and you have to check if the Z1 actually corresponds to cell state. And you cannot dictate, say, I want a cell state representation in Z1 when you are training, because it may not be.
Right. So the interpretability of Z1 would be to some extent a separate question. However, if we can predict perturbed expression, which I think we could maybe argue is cell state to some extent, then I think Z1, we would also know that Z1 has that cell state information. And so that's my thought process.
Okay.
Thanks for the question, though.
Sure.
There is a question in the chat.
Okay. So suppose this resulted in enough drug candidate recommendations that if adopted, you could observe the candidates travel through the pipeline. Would there be a way in the near future to use the data from their journey through the pipeline to improve your model? That's a good question. Yeah, that's a really interesting approach that I haven't considered, because it's kind of that down the line step. I think we could evaluate algorithm performance to some extent if the prioritized candidates have much better, if a higher percentage of them make it through FDA trials, then that would be pretty excellent evidence that our algorithm is performing well. I'd have to think more about how we could use that to actually hone or constrain the algorithm itself. But yeah, that's a very interesting idea.
I guess maybe I can ask a question that sort of gets back to where I thought Shannon was going. Thinking of this from a standpoint of improving the availability, of drugs. What metrics are you using to know that you've succeeded? Is it like more drugs that make it into bench testing, or more drugs that make it beyond bench testing into clinical trials and so forth? Is there any kind of metric that you're thinking about there?
Yeah, another good question. So I think if we identify if we identify these targeted therapies that are selectively cytotoxic to certain cancer cells based on maybe certain mutational patterns or expression patterns, then one, that goes into FDA trials or could go into FDA trials, and that's advantageous. But we can also use this model to try to understand patients who will be more sensitive and therefore select patients for these trials as well. And so we can these targeted therapies are specific to a subset of patients, potentially. However, they will be, I think that they would be more effective for this subset. And so I think we can really boost the treatment outcomes in this way.
Yeah, I think that's probably ultimately where you'd want to go. There's a question by Aaron in the chat box, which I don't know how well you can see. You can share your screen if that'd be easier.
So what are your thoughts about incorporating new drugs into your algorithm? What kind of and how much bench lab data would you need for there to be enough features to generalize to new drugs? This is an excellent question, and it's probably one of the main limitations of my model, because the way I'm encoding drugs, I have to know the protein target and the binding affinity. And for this, there's actually limited data. So Aurora Blicha has done some amazing work in curating both binding affinity and drug target data for oncology drugs, but this is still pretty limited. So for instance, I've done some EDA in this drug data set called GDSC, and there's really only comprehensive drug target data for 10 kinase inhibitors there out of several hundred drugs. And so we actually have a fairly narrow subset of drugs that we can use at this point. But I do think that there's some ways to address this. One, there's a lot of work being done to characterize the binding affinities and the protein targets of oncology drugs. And so maybe in a few years, this will be much better and we can keep adding to it. The alternative route is there's also been a ton of work done in creating computational models to predict drug targets and binding affinity. And so one idea I've been playing with to address this limitation is that we could actually chain, we would basically first predict protein targets and binding affinity for a given drug, and then we could use that in our model. And it makes me a little nervous because we're kind of like chaining the uncertainties from that computational model into the uncertainties with this computational model. However, it would really open the door to the number of drugs we could use at this point. That said, since we're using this this graph-based protein-protein network to constrain feature interaction, I think that this actually will generalize much better to new drugs with smaller data subsets. And so I think that's actually a really that's an advantage of a framework like this. I'm not sure.
Okay, why don't we go?
Yeah,
sorry. Go ahead.
No, I can stop there.
Well, I was just going to say, maybe we can take one more question from the public. There's a question in the chat box about from Mina about assumption of normal distribution of the latent variables. How good is the assumption?
Yeah, that's another great question. So this gets into a little bit of the argument between variational autoencoders and GANs, or there are some other frameworks called, so there's something called an adversarial variational autoencoder, which lets you assume a more complex distribution in that latent space, or something called normalizing flows. But the idea is that can you make the assumption of a more complex function in the latent space? And that's been seen to improve performance in a lot of different tasks. However, it requires more data, and it can affect the interpretability in some ways. And so for at least this first iteration of or development iteration, I want to start with the normal assumption, which I think is fairly fairly valid.
Great. Okay, well, thank you for an interesting talk, which generated a lot of questions. And so we will all leave now, except for your committee and yourself. And then they may have further questions or advice for you. So thanks a lot, everyone, and see you all next time.
Yeah, thank you.